Hi, I'm Jake McClaskey, an environmental specialist from the Bureau of Marine Water Monitoring.
And I am Lisa Dielmo, also an environmental specialist with the Bureau of Marine Water Monitoring. Today we're going to be speaking about the National Shellfish Sanitation Program, NSSP. We're going to be speaking about sampling procedures, sample analysis, and data assessment and reporting for the classification of shellfish waters as per NSSP. New Jersey Administrative Code NJAC 7:12, shellfish growing water classification. The department makes the applicable standards of the NSSP Guide for the Control of Molluscan Shellfish part of the Shellfish Growing Water Classification (SGWC) rules, NJAC 7:12 through rulemaking. In order to protect the health, safety, and welfare of the public from the risks associated with the consumption of shellfish, the SGWC rule establishes the procedures for the classification of shellfish waters and the boundaries of the waters so classified, the procedures for the department to impose immediate shellfish harvest suspensions and restrictions, the requirements for shellfish license holders for the harvest, handling, and transport of shellfish in order to ensure harvested shellfish are safe for human consumption, and permits for aquaculture and harvesting molluscan shellfish from waters other than approved. The National Shellfish Sanitation Program, NSSP, is the federal and state cooperative program recognized by the U.S. Food and Drug Administration, FDA, and the Interstate Shellfish Sanitation Conference, ISSC, for the sanitary control of shellfish produced and sold for human consumption. The purpose of the NSSP is to promote public health protection and improve the sanitation of bivalve molluscan shellfish moving in interstate commerce through federal and state cooperation and uniformity of state shellfish programs. The requirements and recommendations of the NSSP are incorporated into the Guide for the Control of Molluscan Shellfish, which also includes the model ordinance. There are over 1,600 active sampling stations in New Jersey. 88% of these stations have been active for at least 30 years. Our current database only goes back to 1993, so I'd be willing to bet that a lot of these stations are much older than that. Stations are placed where they are likely to see the most influence of pollution from runoff. They are concentrated around the perimeter of a water body, close to stormwater outfalls and the mouths of the tributaries emptying into that water body. As you can see, the stations are located around the perimeter of this water body, but they are also stations leading out into the center to get a good picture of the whole water body. Stations may be created, activated, or deactivated due to changes in the shoreline that make it harder for the sampler to reach the site. New stations may be added if new classification lines need to be drawn.
Prior to sample collection for the NSSP, sampling bottles must be washed and checked for sterility to ensure there is no contamination upon collection of the sample. Bottles are washed in a bottle washer with hot water and a detergent, and then they are rinsed with deionized water. Sampling bottles are also checked with a bromothymol solution to ensure that there is no residual detergent in the bottles, and then they are autoclaved and sterilized at a temperature of 122 degrees Celsius. One bottle from each 32 bottle rack is tested for the residual detergent with aqueous 0.04% bromothymol blue after progressing through a wash cycle. After steam sterilization at 122 degrees Celsius in an autoclave, one bottle per day is tested for sterility using lauryl tryptose broth, which detects the presence of coliform organisms. Results are recorded and records are maintained in appropriate laboratory notebooks. Here are a few pictures of those controls. Wash bottles on the left are dried, capped, and foiled, and then equipped with steam indicator tape. One bottle per each 32 bottle rack is tested for residual detergent with 0.04% bromothymol blue. One glass filtration funnel is tested each day for residual detergent with 0.04% bromothymol blue. The bromothymol blue will change color if there is residual detergent present, so the green is an indicator that there is no residual detergent. Once they pass that quality control, they are placed into an autoclave and sterilized at a temperature of 122 degrees Celsius for 15 minutes. One autoclave sampling bottle from each day is tested for sterility with lauryl tryptose broth. And then sterilized bottles, you can see that the steam indicator tape is turned black. They are dated and set out to be picked up by captains. Sterilized bottles are good to be used for NSSP sampling collection for 1 month. Prior to collecting a sampling run, captains will grab a rack of bottles and label them with the correct number of samples within that run. A temperature blank is also filled with deionized water and included with the sampling bottles for that day. Laboratory analysts on the right, you can see, prepare MTEC plates with the captain's last initial, bottle numbers, and the sample volumes to be filtered from that particular run.
As Jake mentioned, the captains have a quality controlled water temperature blank. They are there to test proper sample temperature consistency. In the middle, we have a picture of the sterilized, numbered, empty bottles to be used for sample collection. They are covered with ice so the bottles don't get too hot while the captain is collecting the samples. On the right, you see a sampling stick that is measured and labeled at 12 inches for correct sampling depth. The sampling site coordinates are programmed into the GPS unit on each boat. According to the EPA, a 37-meter radius around the sampling station is the accepted area in which a sample can be collected. The captain places a sterilized sample bottle into base of sampling stick. He removes the foil-covered cap from the bottle, protecting it from contamination. Sink the sample bottle 12 inches below the surface. Remove the bottle from the water and pour out a small portion to allow for proper mixing of the sample for analysis. He replaces the cap on the bottle and closes tightly, and then places the sample bottle in the ice cooler for preservation. After all the samples have been collected, the captain returns to the dock. In his truck, he has stored a cooler full of ice, which he then covers the sample bottles with for preservation while in transit to the laboratory.
Once the sampling run arrives to the laboratory, the lab staff will check the temperature of the temperature blank to ensure that the samples have been properly preserved. We are looking for a cooling process to have been started, or in the winter months, we are looking for a temperature below 10 degrees Celsius. Captains enter the sample collection start time and their collection intervals into a Microsoft Access database. They then print a laboratory data sheet for each sampling run that they collect. The bacteriology lab is approved by the New Jersey Office of Quality Assurance and a team of FDA laboratory evaluation officers. We employ the MTEC method to determine fecal coliforms in our growing waters around the state, and we classify based on that fecal coliform standard. The laboratory is subject to audits performed by both the NJDEP Office of Quality Assurance as well as the FDA's team of laboratory evaluation officers. The Bureau of Marine Water Monitoring strictly adheres to the requirements set forth in the FDA checklist, and the checklist allows, again, for the use of membrane thermotolerant Escherichia coli agar, MTEC, for the bacteriological analysis of seawater. So each day, the laboratory staff perform quality controls on anything that may come into contact with an NSSP sample. So pipettes are washed with a detergent, they are rinsed with hot tap water and then deionized water, and sterilized in a hot air oven for 2 hours at a temperature of 170 plus or minus 10 degrees Celsius. Sterility for each batch of pipettes is confirmed with a dry heat indicator strip and double strength tryptic soy broth. The temperatures of all required laboratory equipment are recorded twice a day, morning and afternoon, to ensure that they are within the required range. Again, sample collection bottles are washed and tested for residual detergent, and then they are tested for sterility after being autoclaved with lauryl tryptose broth. Positive and negative controls are prepared from stock cultures for each ethafoam container, which you will see at a later slide, containing NSSP samples in the 44.5 degrees Celsius incubator. Phosphate buffered saline is used as a rinse solution. The saline is autoclaved and tested for sterility by dispensing it into double strength tryptic soy broth before use. Saline sterility is also checked each day that samples are analyzed with a tryptic soy agar rinse plate that each analyst performs prior to sampling, or prior to filtering samples. Temperature blanks were mentioned earlier. Each captain carries a temperature blank containing deionized water that is placed in their cooler along with their samples. Immediately upon the arrival of samples to the laboratory, a laboratory analyst measures and records the temperature of the bottle. The temperature must be less than that of the growing area waters or below 10 degrees Celsius. Results are recorded and records are maintained in the appropriate laboratory notebooks. Each batch of newly prepared media, so in the case of NSSP samples, the MTEC agar, 4 control plates are set aside to test the pH and to run blank, positive, and negative control plates. Additionally, upon opening, each new lot of media is tested with a blank, a positive control plate, and a negative control plate. All membrane filtration glassware is soaked in hot water with a detergent for at least 1 hour. After that, they are rinsed with hot water and deionized water and allowed to dry. Filtration glassware is then tested with 0.04% bromothymol blue for residual detergent and autoclave for 15 minutes at 122 degrees Celsius for sterilization. Each day, laboratory analysts perform their own set of controls prior to filtering samples. Each laboratory analyst tests the sterility of their filters and phosphate buffered saline on a tryptic soy agar plate. One additional tryptic soy agar plate accompanies the control plates for the day to verify the sterility of the media. Each laboratory analyst also performs an MTEC start plate prior to filtering any samples, and also performs an MTEC end control plate at the conclusion of filtering the day's samples. In between the MTEC start and end control plates, each analyst also performs an every 10 control plate for each time 10 samples have been filtered through a glass filtration apparatus to ensure that they are performing a proper rinse in between samples. One MTEC media blank plate also accompanies the day's samples to verify the sterility of the media. Each week, the temperature of each autoclave is measured and recorded with a maximum registering autoclave thermometer. Stock bacterial cultures are transferred to new broths to ensure proper growth. New positive control cultures are prepared, and all media and sterilized sampling bottles are checked to ensure that they are within the acceptable date range. If bottles happen to expire outside of that 1 month limit, they are rewashed and re-sterilized, and all expired media is discarded. Each month, field and trip blanks are analyzed to check for contamination that may occur during sample collection, preservation, and transport. A trip blank and a sterile water bottle, each filled with sterile deionized water, are provided to each captain at the beginning of each month, along with an empty sterile bottle labeled field blank. During a sampling run, captains transfer the contents of the sterile water bottle into the empty field blank bottle, thereby simulating the collection of a sample. The trip blank bottle remains unopened in the cooler throughout the entire sampling run. Upon arrival at the laboratory, the field and trip blank bottles are tested for the presence of coliform organisms by dispensing 5 10 milliliter aliquots from each bottle into sterile tubes containing 10 milliliters of double-strength lauryl tryptose broth equipped with Durham tubes. After 48 hours of incubation, the tubes are checked for gas production resulting from the fermentation of lactose by coliform organisms. If no gas is present after 48 hours of incubation, the field and trip blanks are deemed to be sterile, ensuring no contamination has occurred during the sampling process. Here's a picture of a laboratory analyst analyzing the captain's field and trip blanks. Each month, the air quality of the laboratory is tested by using a large loose-fitting plate containing standard plate count agar that is left exposed in the laboratory for 15 minutes and then incubated at 48 plus or minus 2 hours in a 35 degrees Celsius incubator. The resulting colony count is recorded and records are maintained. Each balance used by lab staff is also checked monthly according to the manufacturer specification using National Institute of Standards and Technology, or NIST, Class S1 weights. The accuracy of the balance is verified at the weight range of use. Results are recorded and records are maintained for the balances. The dry heat oven that we use to sterilize our pipettes is tested by using a live culture strip that is placed in the oven with a load of pipettes. The strip is then placed into tryptic soy broth to confirm sterilization. A positive control strip is also incubated along with the autoclaved strip. The deionized water storage tanks are tested monthly and must be higher than 2.0 megohms per centimeter of resistivity. Storage tanks are tested monthly using the heterotrophic plate count pour plate method and must contain less than 100 colony forming units per 1 milliliter of deionized water. UV sterilizers are tested monthly using a large loose-fitting plate containing plate count agar that is inoculated with a positive E. coli control. The plate is placed in the UV sterilizer for 2 minutes and then incubated for 48 plus or minus 2 hours in a 35 degrees Celsius incubator. Results should be negative. If a plate shows positive growth, the sterilizer has maintenance performed on it and the test is repeated. Autoclaves are tested monthly for sterilization effectiveness using spore ampules containing Geobacillus stereothermophilus spores. After sterilization, ampules are placed in a water bath and along with a positive control that is not autoclaved for 48 hours. The spore that went through the sterilization cycle should show no growth, and the positive control should show growth. Duplicate data. Samples are run monthly for precision analysis by each laboratory staff. Lab analysts rotate the analysis monthly, and records are documented in both the laboratory notebook and electronically. Duplicate plates for MTech must go into the same ethafoam container along with a negative control. Each year, the laboratory performs thermometer calibrations using a proper NIST certified thermometer. We send our laboratory water samples to the Department of Health for metals analysis. We analyze laboratory tap water for total coliforms. The laboratory supervisor reviews the FDA checklist annually to ensure the laboratory is in conformance. A copy of the checklist stating any nonconformities and corrections is documented and kept with the laboratory standard operating procedure within the laboratory. Laboratory proficiency testing is administered annually by the Food and Drug Administration to comply with all NSSP model ordinance criteria for the control of molluscan shellfish. All laboratory analysts participate in the annual proficiency testing. Records and results are maintained by both the laboratory and through the FDA's Food Shield program online. Analyzing an actual sample. Bottles are vortexed for 15 seconds prior to filtration to ensure that the sample is properly mixed. A 3 milliliter sample volume is pipetted into a sterile filtration apparatus, and a pump is used, a vacuum pump is used, to pull the sample through a sterile filter. A 30 milliliter sample volume is then poured into a sterile filtration apparatus, and a pump is used to pull the sample through a sterile filter. Here's a video of a laboratory analyst filtering a NSSP sample. Forceps are sterilized using a flame. This filter is placed onto the filtration apparatus. A second filter is dispensed and placed onto a sterile filtration apparatus. Roughly 20 milliliters of saline is dispensed to ensure proper dissolution of the sample within the 3 milliliter aliquot. The sample is vortexed for 15 seconds to ensure proper mixing. The sample is opened and a sterile pipette is drawn from the canister. A 3 milliliter aliquot is drawn into the pipette and dispensed into the saline. And then a 30 milliliter aliquot is poured to a point that is marked on each filtration glassware. The samples are pulled through the filter so that all the bacteria within the sample is trapped on that filter. And then two 20 milliliter rinses are performed to ensure that all bacteria that may have been stuck to the side of the glassware is onto the filter. The forceps are then re-sterilized with a flame. The laboratory analyst picks up the filter and places it onto the MTech auger, ensuring that no air bubbles form between the filter and the auger itself. And the same is done for the 30 milliliter aliquot. The filter, now containing all fecal coliform bacteria from the sample, is placed onto a sterile petri dish containing MTech agar. The samples are then loaded into sealed ethafoam containers to slow the temperature ramping process during incubation. Inoculated MTech plates are incubated at 24 plus or minus 2 hours at 44.5 degrees Celsius to allow for optimal resuscitation and growth of fecal coliform colonies. Incubation dates and times are recorded on the bacteriological analysis worksheet that the captain provided upon collecting his run. On the left, you can see a filter that has been placed on the MTech agar that has been incubated, showing the growth of fecal coliform colonies after incubation. All yellow, yellow-green, or yellow-brown colonies are counted as fecal coliform colonies when scoring plates. A laboratory analyst you can see in the middle is counting and recording the fecal coliform colonies that grew during incubation. Counted colonies are recorded on the captain's data sheet, and a result is then calculated and reported in colony forming units per 100 milliliters. Once a captain's sampling run is filtered, incubated, and scored on the bacteriological analysis worksheet by a laboratory analyst, a separate laboratory analyst then reviews the calculations to ensure that they are correct based on colony counts and volumes filtered. Once a sampling run is scored and the results are reviewed, the results are entered into a Microsoft Access database to be analyzed and used by report writers to update existing classifications. Prior to the data being made available to report writers, it is reviewed by a separate laboratory analyst to ensure that the results were entered into the database properly, thus eliminating the possibility of transcription errors. On the right, you can see a complete bacteriological analysis worksheet with incubation start and end times, the data entry date in the top right, and the data validation date just below it.
There are two sampling strategies used for shellfish sample collection. The first is the systematic random sampling strategy, or SRS. This is used in areas where non-point pollution occurs, as it is less predictable and occurs under different conditions. The sampling plan is set up ahead of time so the conditions are random and are collected under different circumstances, such as rainfall and water temperature. At the end of each calendar year, a new SRS sampling schedule is created and handed out to each of the captains so that they can plan their sampling year accordingly. A minimum of 6 samples are collected per year for this sampling type, and shellfish classification is based on the most recent 30 samples, usually around 5 years worth of data. Data for this sampling strategy is assessed using the estimated 90th percentile and the geometric mean. The other sampling strategy is called adverse pollution control, or APC. This one is used in areas where point source pollution is more prevalent. Samples are collected under conditions that historically result in higher fecal coliform levels, so they are not scheduled ahead of time. In New Jersey, these tend to be rainfall and seasonal conditions. A minimum of 5 samples per year are collected. Shellfish classification is based on the most recent 15 samples, usually around 3 years worth of data. Data is assessed using the geometric mean, and 10% of samples cannot exceed criteria. The estimated 90th percentile shall be calculated by calculating the arithmetic mean and standard deviation of the sample result logarithms, base 10, multiplying the standard deviation in A by 1.28, adding the product from B to the arithmetic mean, taking the antilog base 10 of the results in C to get the estimated 90th percentile. The geometric mean is defined as the antilog base 10 of the arithmetic mean of the sample result logarithm base 10. These calculations are performed by total access statistics. Under the NSSP, shellfish growing areas may be classified based on their fecal coliform results in one of the following ways. First, we have approved, an area where harvest of shellfish for direct marketing is permitted. Prohibited, an area where the harvest of shellfish for any purpose is not permitted. Conditionally approved November, an area where harvest for direct marketing is allowed only from November through April. Harvest during all other times of year is by special permit only. Conditionally approved January, an area where harvest for direct marketing is allowed only from January through April. Harvest during all other times of the year is by special permit only. Restricted, an area where harvesting shall be by special permit, and the shellfish following harvest is subjected to a suitable and effective treatment process. And lastly, we have suspended, an area that is temporarily closed for harvest. For the data assessment, I'm going to be using the Tuckerton Creek area as my example. Here is a visual of that area. Once the data has been added into our database, I can then, as part of the assessment team, can go in and look in total access statistics and to ask it to run all different scenarios for me. So the first thing I see is on the left where it says bacterial analysis. This is where I choose all the different aspects of the reports I want to see. I can, on the left, you see I have checked limit data set. You put an end date in there and it will count back the most recent 15 or the most recent 30, depending on the sampling strategy. You can also uncheck limit data set and you can pick your own start date and end date. You then choose your report area. New Jersey is broken down into 24 different report areas. So here's where I break it down to which report area I'm looking at. Then we can choose the active or inactive stations. We can also limit the stations. Once we have chosen a report area, if we hit limit stations, it will just show us the stations in that area. So we can choose, maybe we've seen one that is doing exceptionally well, or maybe isn't meeting criteria. We can limit the stations and just choose the data for those stations. We also can choose our wet dry cutoff criteria. 0.2 inches is the generally accepted amount of rainfall for this. However, you can run all different scenarios, any amount, 0.1, 0.3, whatever you choose, and then you can compare different rainfalls to each other. So once I choose the scenario that I want, I click get results, and on the right, this is what I see. It's asking me now which reports I would like to see. So I choose raw data. That will show me the raw data for all of the stations in the area, if that's what I've chosen. I choose the sampling strategy that is for that growing area. And then I choose, I want to see my seasonal influences and all of my rainfall influences from 1 hour, 3, 6, 12, 24, 36, 48, 72 hours prior to sampling. And then I also want to choose my rainfall amounts. That will give me the rainfall for all of those rainfall times for every date that a sample was taken. These are the reports that I just chose to get on that last screen. So on the left, it gives me the stations that I chose, and it's comparing them to approved criteria. So as you can see, none of the exceed criteria is checked, so every station in this area is meeting approved criteria. If one wasn't, then that would be checked, and then I would look into that one further. On the right, it's now comparing it to restricted criteria because there are a couple of restricted stations in this area. As you can see, all of those are also meeting restricted criteria. On the bottom, we have, now I'm looking at the conditionally approved November stations in this area. For those, because I need the 30 most recent data points, I need the 30 most recent data points in winter when this station is open for harvest. So I choose just the conditionally approved stations and I change my data time frame. So for this, I had to go back to March of 2019 to get my 30 data points in the winter. As you can see, these stations meet approved criteria year-round and also during the winter when the station is open for harvest. Our data is assessed using a revolving data system. When a new data point is added to the database, the oldest data point is taken away and a new estimated 90th percentile and geomean are calculated. If you look on the left, we go from September of 21 through June of 25. Well, on the right, you see that now we have added another sample on July 17th of 25. So this sample from September 20th, 21 will now go away. And now these 30 samples will be recalculated for the 90th percentile and geomean. You can see that the 21 from September of 21 was replaced by a 33. So the estimated 90th did go up just a little bit. If you take a look at September 8th of 2022 on the right, you'll see that it's a 58. That isn't exceedingly high, but for this data set, it is. So now I will go to my rainfall summary and look at that date, which was July 8th, 2022, and see that 48 hours prior to sampling, there was 3 inches of rain. So this may or may not have an influence on this station, but it is something that I check and will look into further. On the right, you can see that I chose 48 hours as my example for the cumulative rainfall because this is where this area had the most rainfall influence, most stations with rainfall influence. Just because a station has a rainfall influence does not mean that it does not meet criteria. The test to determine the wet-dry statistics is called a t-test. It is a statistical test that is used to compare the means of two groups. We also do a seasonal analysis. It calculates the summer geomean and the winter geomean, and it compares them, also using a t-test. Generally in New Jersey, our stations that do have a seasonal influence will have a summer seasonal influence, and that is because there is higher populations, more boat use, and higher air temperatures in those dates. All of this data assessment accumulates into these three different types of reports that we write. First, we have the annual report. Obviously, that one is conducted annually. The data is reviewed to see if there's any areas of concern. Each of the team, the assessment team, goes and looks at their respective growing areas to see if there are any areas of concern or areas that maybe could be upgraded. We then meet with our bureau chief. We meet all together with our bureau chief and our FDA shellfish specialist to discuss what happens. An area of concern could be where a station is already not meeting criteria, or just an area that is maybe trending towards not meeting. The stations that are not meeting criteria currently will be suspended in the interest of public health, and the other areas where it still meets criteria but it's trending towards not, we will then maybe do some special projects, do some potential source track down projects, and look into the issues to see if we can make any fixes as to prevent the area from being downgraded or suspended. The next report we write is a reappraisal report. This report is written every 3 years, and it's more in depth. We add a shoreline survey aspect to this report. We go to each of the marinas in the growing area. We look to see if any changes have happened in, say, the footprint of the marina or the size boats that the marina can hold, different owners, that any changes that have happened in the most recent 3 years. The data that we get from these marina surveys is used to calculate a buffer around the marinas. So the amount of slips and size of boats is something that we use in that calculation. So if there were changes, a new marina buffer will be calculated. We're also looking for new construction, anything that might have changed in the area since the last time we were there. In this type of report, we can make classification downgrade changes. We recommend downgrades in this growing report based on the shoreline and the data. The next type of report is the sanitary survey report. It is conducted every 12 years. This one is much more in depth. We do much more shoreline survey. It encompasses more details such as new pollution sources, new stormwater outfalls, septic system locations and maybe malfunctions, updated land use, and we visit the wastewater treatment plants. This report is where we can recommend upgrades. Upgrades require obviously the data to meet criteria, but also extended shoreline survey to back up that data. So we can use this report as a way to upgrade our waters. The FDA issues reports annually and biannually assessing the programs that implement the NSSP. Concerns that the FDA may have about program implementation are noted as emerging concerns, and remedial action is outlined. If remedial action is not successfully implemented, the programs can be found to be in noncompliance with the model ordinance. If states are found to be in noncompliance with the model ordinance, the interstate sale of shellfish harvested from New Jersey waters can be stopped. If you have any questions about this presentation, please feel free to reach out to any of these people listed on your screen at any time, or you can call us at the office at 609-748-2000.